Welcome to this explainer. Okay, today we're doing something pretty incredible. We're going to shrink right down and embark on this dynamic, microscopic tour of a cell to explore its internal structures. I want you to imagine a bustling, microscopic city, because that's exactly what we're going to map out together right now. Let's get into it.
We're starting with this foundational premise straight from the chapter text. It says, being the structural and functional unit of a living organism, cell has got a very important place in understanding the entire functioning of a living system. And honestly, that grounds us in exactly why understanding these tiny cities is so absolutely crucial. To figure out how a massive, complex living system actually works, you have to start at its smallest fundamental unit. We're looking at the literal engine of life itself.
So here's our tour map for today. First, the cell's dynamic boundary. Then, ribosomes, protein synthesizing factories. Third, the endoplasmic reticulum. Fourth, power and metabolism centers. And finally, we'll hit the nucleolus command center.
Let's kick things off with part 1, the cell's dynamic boundary. Our very first stop is the plasma membrane. Think of it as the cell's border patrol. It guards the cytoplasm and dictates how the cell interacts with the outside world. Now, the way we understand this boundary today is through what's called the fluid mosaic model. Singer and Nicholson proposed this back in 1972, and wow, it was a real game changer. It showed us that the membrane is not some rigid brick wall, far from it. It's this dynamic, shifting lipid bilayer studded with a gorgeous mosaic of globular proteins. It's fluid, and it's constantly in motion.
But what's really fascinating here is the actual chemical makeup. You'd probably assume a lipid boundary is, well, mostly lipids, right? Actually, it totally varies by cell type. To put it into perspective, if we look at human erythrocyte membranes, those are your red blood cells, they're packed with more proteins than lipids. We're talking 52% protein to 40% lipids. Kind of wild when you think about it.
Let's zoom in a bit closer on those lipids. The main ones are phospholipids, and they arrange themselves so perfectly. They've got these water-loving or hydrophilic heads that face outward and water-fearing, hydrophobic tails tucked safely away in the interior. This specific setup creates a quasi-fluid state, and because it's so fluid, those proteins we just mentioned, they can literally float and drift laterally right across the cell boundary.
Moving on to part 2, ribosomes, the protein synthesizing factories.
All right, so we've crossed the border into the cytoplasm, and right away we bump into the busy factories of our microscopic city, the ribosomes. These things are incredible. George Paley first spotted these little workers way back in 1955 using an electron microscope. And what's super unique about them is that they're entirely membraneless, no protective wrapper or anything. They just sit there, scattered all over the cytoplasm in both prokaryotic and eukaryotic cells, continuously churning out proteins.
Now, when you're reading through the chapter, you'll constantly see these ribosomes categorized by a number with the letter S next to it. That S stands for a Svedberg unit. It's named after the Swedish chemist Theodor Svedberg, who, by the way, actually won a Nobel Prize for inventing the ultracentrifuge. But here's the kicker. The Svedberg unit isn't a measure of weight. It's a measure of sedimentation rate. Think of it like a speed limit for how fast the particle sinks when you hit it with an intense gravitational field inside a centrifuge.
The key takeaway here is that ribosomes come in slightly different models depending on those sedimentation speeds. Your standard eukaryotic ribosomes scattered around the cytoplasm, those are your 80S models. But if you peek inside certain organelles, like chloroplasts, or if you look at simpler prokaryotic cells, you're going to find the slightly smaller 70S version.
Let's dive into part 3, the endoplasmic reticulum.
As we float further inward from those free-floating ribosomes, you start to see how the cell's geography really builds. Look at how this massively folded, interconnected network of the endoplasmic reticulum just tightly hugs the nucleus. And if you look closely at its outer surface, you'll see it studded with those exact same little ribosome factories we just toured. This whole sprawling structure acts as a major manufacturing and transport hub for the cell.
But it's not a one-size-fits-all operation. It's absolutely vital to distinguish between the two zones here. The rough ER is the part covered in ribosomes, which gives it that bumpy look. Its main gig is synthesizing and directly transporting proteins. But right next door, you've got the smooth ER. It's completely clear of ribosomes and handles totally different tasks, like lipid metabolism and storing calcium. It's all about highly specialized manufacturing zones.
Which brings us to part 4, power and metabolism centers.
Navigating deeper through the cytoplasm, we run into these specialized metabolic centers that keep our city running smoothly without getting buried in waste.
Now, everyone knows about mitochondria, the powerhouse, right? But let's focus on peroxisomes for a second. These are small, membrane-bound organelles, and they actually derive directly from the endoplasmic reticulum we were just looking at. They literally just pinch right off to go do their specific jobs.
So what are those jobs? Well, they serve as a critical hub for enzymes and are deeply involved in the cell's energy metabolism. And here's a really fascinating point of comparison from the text. Unlike mitochondria or chloroplasts, which have their own unique DNA, peroxisomes completely lack their own genome. Yet, much like mitochondria, they still manage to replicate themselves by fission. Pretty cool, right?
And finally, Part 5, the nucleolus command center.
We have reached the deepest part of our journey inward. Past the nuclear boundary, nestled safely within the nucleus itself, we find the nucleolus. This is a distinct command center that actually, much like our ribosomes earlier, completely lacks a membrane of its own. It's essentially this incredibly dense region that compartmentalizes some highly important nuclear processes.
Its main function? Absolutely critical. The chromosomal region occupied by the nucleolus is just packed with genes that synthesize rNRNA. It acts as the nucleolar organizing region. To put that in plain English, this is the factory that builds the factories. This is exactly where the ribosomes themselves are created before they get shipped out into the cytoplasm to build proteins. It is just a beautiful, full-circle microscopic system.
Well, to wrap up our explainer journey today, let's do a quick chapter knowledge check. Test your memory right now. Think back to our very first stop. Do you remember who defined that dynamic, shifting boundary of the cell back in 1972? Was it A. Robert Brown, B. Sliden and Schwann, C. Robert Virchow, or D. Singer and Nicholson? If you were guessing D right now, you are spot on. Singer and Nicholson hit the nail on the head. Understanding these organelles, how they divide labor, how they build off one another, and how they keep the whole system alive is the crucial first step to understanding all of biology. Thanks for exploring this microscopic city with me. As you go about your day today, I want to leave you with this thought. What other incredible, hidden universes might be operating inside you right at this very moment?